The focus of this video is microfilaments. Here we're watching the crawling movements of an amoeba as it hunts for food. Notice how the cytoplasm is rearranging itself, pulling the cell forward in some regions and retracting the trailing edge in other regions. These cytoplasmic rearrangements are made possible by microfilaments. Microfilaments are one of the three classes of cytoskeletal elements, the other two being microtubules and intermediate filaments. They play critical roles in the cell. We can broadly divide these into two categories. The first is movement. So microfilaments play a role in whole cell motility, like I just showed you with the amoeba, but also in white blood cell crawling, in wound repair, and in muscle contraction. And they also play a role in intracellular motility, dividing the membrane in cytokinesis, moving vesicles and organelles around the cell. The second broad function for microfilaments is structural. Microfilaments play a role in determining the cell shape and providing structural support for things like the microvilli in epithelial cells and growth cones in developing neurons. Staining cells for microfilaments shows that there's normally a dense network that lies underneath the cell membrane. We call this the cortex. So in these two cultured cells, the microfilaments have been labeled red, and then the nucleus is green. Here's an electron micrograph again showing the edge of the cell at the plasma membrane, and then the dense area labeled cortex. That's a meshwork of microfilaments lying right underneath the cell surface and a little bit different from the rest of the cytoplasm. Actin can also be arranged into other specialized structures such as microvilli or adhesion belts, and sometimes actin gets arranged into short-lived transient structures such as the contractile ring. Like other cytoskeletal elements, microfilaments are polymers of building blocks. The building block for microfilaments is G-actin, globular actin. In humans, there are 6 different actin genes, and beta and gamma are expressed in all cells, although they play slightly different roles. And the other 4 actin genes are muscle-specific. Actin is one of the most abundant proteins in the body. It makes up 10% of protein by weight in muscle cells, 1% in other non-muscle cells. Here's a structural model showing us what actin looks like. You can see it's a globular protein. It looks a little bit like a cup or a bowl, and there's a pocket in which ATP can bind. I'm going to move this over, and now I'll show you my cartoon version of what an actin monomer will look like. The two sides of G-actin look different. There's the side that can bind ATP and the side that doesn't. And these are sometimes referred to as the minus side and the plus side. It has nothing to do with charge, but instead of how it's going to behave when it gets incorporated into a larger filament. Another little bit of terminology is that the minus end is associated with the pointed end of microfilaments, and the plus end is associated with the barbed end. Let's look at microfilament assembly. We begin with G-actin monomers, and on their own, they don't have a high affinity for one another. But if they bind ATP, then what happens is you'll get a small cluster, first 2 and then 3 actin monomers will come together. We get what's called nucleation. And from that nucleation site, the actin will grow out. I've added the plus and the minus ends again, just to show you how this happens. So here's our nucleation cluster again, and what will happen is individual monomers will hop on, elongating the microfilament. Now a couple things about my cartoon. Although I've colored the subunits a light blue and a dark blue, that's just so you can keep track of where they are. They're identical subunits. The other thing you might have noticed is that I've shown the monomers preferentially adding to the plus end. That's what the plus end means. It means it grows more rapidly than the minus end. There are some actin monomers that can join onto the minus end, but the rate of addition at the minus end is much slower. And what that means is that as these clusters elongate, they grow preferentially from the plus end. The resulting structure looks like 2 strands coiled around one another. We call this filamentous actin, or F-actin, to distinguish it from the globular actin. Well, here again is another cartoon of my microfilament, and again I've drawn the 2 strands with 2 different colors, simply so you can see where they are. And remember that the monomers are preferentially hopping on to the barbed end, the plus end. It turns out that actin has an ATPase activity. So with time, ATP is hydrolyzed, first to ADP and inorganic phosphate, and then it releases the inorganic phosphate to form ADP. And once the monomer is bound to ADP, it has a low affinity to stay in this configuration. If we look at our filament now, remember that most of the new monomers are being added to the plus end, so the oldest monomers will be near the pointed end. That's where the ATPase activity will have led to monomers with ADP, and those will tend to dissociate. This leads to a phenomenon called treadmilling, where when we're looking at actin in the cell, even if a particular length of actin doesn't look like it's getting any longer or shorter, there may be continual turnover as new monomers with ATP join onto the plus end and monomers with ADP fall off the minus end. These events are also shown in this cartoon, where the monomers bind ATP to nucleate and then polymerize, and then with time, ATP will be hydrolyzed and there'll be depolymerization. Well, any process like this where ATP is being consumed is costly to the cell, and so you won't be surprised to learn that cells have ways to regulate this. Cells use a number of proteins to regulate the assembly and disassembly of actin filaments. For example, there are some actin-binding proteins which will promote nucleation. This is a rate-limiting step in the initiation of a microfilament, so adding a nucleator will speed this process up. There are also proteins that can sequester the subunits. If we just took a bunch of actin monomers and ATP and put them in the test tube, they would be pushed towards polymerization. But in the cell we find that there's a great excess of unbound monomers, and that's partly because they're monomer sequestering proteins which can hold the globular actin out of the pool until it's needed. There are still other actin-binding proteins, some of which will cap the ends so that no more microfilaments can be added. Some will cross-link or bundle them. There are some that can sever the filaments if they get too long, and there are some that will be associated with binding to the membrane. Collectively, these are called the actin-binding proteins, or ABP, and there are a lot of them. More than 100 exist. They can affect the localized assembly or disassembly of actin, their physical properties, and their interactions with one another and with other organelles. And there are many different subtypes that carry out different functions. Modulation of the actin cytoskeleton by actin-binding proteins can help explain changes in cell shape like that amoeba crawling. Let me show you what I mean by this. Here's an assay that's used to study cell movement in cultured cells. Can plate cells out so that they form a monolayer and then scratch, forming a lane where there are no cells left. Over time, the cells rapidly change shape and grow in to close this space. It's a model for wound healing. If we add drugs that block actin polymerization or that affect some of these actin-binding proteins, this process can be stopped. The model to explain the crawling motion, like in the amoeba or in this wound healing, involves controlling the polymerization so that the cells preferentially add actin monomers close to the membrane, pushing them out, and then they sever and they depolymerize the older elements away from the edge of the membrane, so that the cell is continually making the building block, these actins with ATP bound to them, in order to push the membrane out and allow the entire cell to change shape. This type of motility, crawling motility, doesn't involve a molecular motor. There is, however, a molecular motor for microfilaments. Its name is myosin. Myosin is a superfamily of molecular motors. There are many members. It's best known for its role in muscle contraction, but it's also involved in intracellular motility of organelles and vesicles. Humans have about 40 different myosin genes in 12 different classes, and presumably these are carrying out different functions in different cells. A single cell might express multiple forms of the myosin, which they can use for different tasks. In microtubules, we saw that there were two different kinds of molecular motors, kinesin and dynein, one that walked towards the plus end and one that walked towards the minus end. But almost all myosins, there's only one exception, walk to the plus end of actin. So when you think about myosin, think about it as being a plus-directed motor. Here I'm showing you a cartoon of some of the different types of myosins, and I want you to notice that they don't all look alike. There are some differences. Despite this, they do have some similarities. All of them are composed of either one or two heavy chains. That's what's shown in the turquoise. And then there are several smaller, light chains. Those are shown in green. Let me blow this up just so we can look at them in a little bit more detail. What I want you to notice is that although the myosins look different, each one of them has a conserved head region with a little cleft in it. That'll be the binding site that can interact with actin, and it will also be the site where ATP will be hydrolyzed in order to allow the motor to take a step on the myosin. The tail region's more variable. That's what's going to be associated with cargos. Some myosins, and we see this with myosin 2 and myosin 5, can form homodimers, where the tails wrap around one another. We call this a coiled coil motif because there are alpha helices in the tails and then they coil around one another. Cell biologists will often divide myosins into two groups. Myosin 2, the conventional myosin, that includes muscle, which is the best understood, and then all of the other myosins will be considered non-conventional myosins. Those aren't involved in muscle contraction and instead are associated with cargo. I want to close this video by telling you a little bit about what's arguably the best understood of the unconventional myosins, Myosin 5. Here's a cartoon version of Myosin 5. Actually, there are two cartoon versions. On the left, we just have what it would look like with the globular heads in red and then the tail region in green. The light chains for this myosin are called calmodulin. They're shown in blue here. On the right side of the figure, you can see Myosin 5 associated with actin. That's on the bottom, and notice that the globular heads are interacting with the actin. And then the tail end is interacting through a couple adapter proteins to its cargo, which here is labeled melanosome. Myosin 5 serves a number of different functions, but one of them is moving pigment granules into what will become the hair. So here's a mouse model for a mutation in Myosin 5. Normally the mouse has a dark fur color, but mice that have a mutation in the Myo5A gene are unable to transport the pigment as effectively, and they have this washed out or diluted fur color. Now I want to zoom in and look at how the myosin heads are interacting with the actin filaments. That's what we're seeing in the bottom. Myosin 5 is one of those myosins that's a dimer. There are two globular heads. Here they're shown in different colors, the turquoise and pink. They're identical, but that's just to help us keep track of them. What I want you to know about the way that myosins walk along actin is that it's going to involve binding ATP and breaking it down and using that in order to take a step. A general rule, not just for Myosin 5, but for myosins in general, is that the globular head has a high affinity for actin when ADP, or no nucleotide, is bound to that cleft in the globular head. But if they have ATP, or if the ATP has been hydrolyzed to form ADP and an inorganic phosphate and both are bound, then the globular head will have a low affinity. So what we're looking at then in the bottom figure on the left is the two heads both have ADP bound to them, and because of that they have a high affinity and they'll be associated with the actin. When the ADP is released, the turquoise head still has a high affinity for it, but the moment that that turquoise globular head binds ATP, it will let go of the actin, and then as it hydrolyzes it, that's going to provide the energy in order to do a conformational change and swing that back leg forward, so now it's in the front. When the inorganic phosphate is released, so that the globular head only has the ADP bound to it, now it has a high affinity again, and it will step down, and the globular head will bind to the actin again. So each time the molecular motor takes a step, the ATP will be hydrolyzed, there'll be a conformational change, and it will move one step forward. As I said before, Myosin 5 is an example of an unconventional myosin, and these play a lot of different roles in the cell. They're involved in moving the melanosomes, but also in things like phagocytosis and endocytosis. There are interactions between unconventional myosins and the membrane that help shape the microvilli and stereocilia. And these myosins play a role in other processes in the cells, such as autophagy or organelle localization. Although there are many different myosins, the similarity in the globular head means that the basic principle of how the ATP will interact with the actin will be shared across these myosins. It's also shared in the case of muscle myosin, that conventional myosin, but muscle contraction is a complicated process, and I think we'll leave it for another day.